for coming. People in Zoom and people in person. I appreciate you guys all I'm glad that you all are here. We have Spencer here from Soul Mushrooms and Lion Farms, and he's gonna go ahead and talk about the mushrooms he's growing at his farms. So without further ado, Spencer.
Thank you guys. Appreciate you having me here today. So I'm the founder and cultivator behind Soul Mushrooms. I started this company a little under 2 years ago. There weren't a lot of mycologists on the scene here, so basically self-taught. I don't have a science background of any kind, but YouTube is an amazing tool to use. And the internet, and then trying to connect with other mycologists in other cities and states to learn the process, and then really it comes down to trial and error. That's the best way to learn, is by failing. So when I started this company, I had intentions on mostly growing the gourmets, but the inspiration came behind the functional mushrooms. Functional meaning they're medicinal. They all have their specific function. They have overlapping functions. So the primary mushrooms we focus on are reishi, cordyceps, turkey tail, lion's mane, and chaga. We don't grow chaga. If you're familiar with that mushroom, I'll share a little bit about it, but it's not a mushroom that can be cultivated. It's not really even considered a mushroom, but still a fungus of some kind. And then the gourmets, so oyster mushrooms, a variety of those, chestnuts, piopinos, and lion's mane. We grow them in different environments, and it allows us to express creativity through the cultivation process, get higher yields, and keep the mushrooms happy. They're extremely finicky organisms. The mushrooms grow with bacteria in nature. They have for eons. But once you take them out of their natural environment, they cannot withstand any type of contamination. Their immune system is not quite the same. So creating the perfect environment, sterile environment, and working in the laboratory is extremely crucial. Otherwise, you'll get some type of contamination. And the cultivation process starts with a petri dish or a liquid culture, and there's many ways to skin this cat, as they would say. So I started by cloning mushrooms that I liked. You can go to the store, you can hit it with isopropyl, you can slice it open, get a sterile scalpel, put a tissue sample of that mushroom on sterile agar. It's very simple. It's agar, reverse osmosis water, and some type of nutrient, which is essentially sugar. So light malt extract is what we use. And after we pour the plates, we let them chill, wait to see if there's any contamination. Most of the time they aren't if you're doing proper sterile procedure. And then I can put a tissue sample on here. I could put a liquid culture of the mycelium on there, and it will expand out and grow. And that starts the production process. And the next step is going on to sterile grains. So there's a lot of grains you can use. We use oat, just organic oat. It's essentially horse feed. We soak it for about 24 hours. We let it air dry, and then we sterilize it in an autoclave for about 2 hours. Once it cools down, this agar or a liquid culture is going to be inoculating that grain. And after a month and a half, the grains get fully colonized by the mycelium, and then it goes into the next stage, which is substrate. And the substrate is also organic matter. We use oak and a little bit of soy hull, which is supplementation, the outer shell of a soybean. So we're working with saprophytic mushrooms that break down dying and decaying matter like trees and logs. There's also the mycorrhizal, which play a massive role in our ecosystem and with the plant life. So as you already know, plants breathe CO2 and release oxygen. And mushrooms, they breathe oxygen and release CO2, creating a symbiotic relationship between the two. So without mushrooms, plant life would not sustain the same way and vice versa. And the name soul came from the acronym seed of life, which is a symbol in the O there, because I envision mushrooms as seeds of life here on Earth. Without that, life would not properly sustain. It's the first organism on here, arguably the most important organism on our planet. And without mushrooms, there'd be a complete imbalance in life here. It's also believed that mushrooms might not have originated here. The concept of panspermia, where potentially life started in outer space or another planet, and meteorites essentially hit our planet. They had water content in them, and that is possibly where life started. And it's believed that mushrooms are potentially extraterrestrials. We're talking billions of years ago. So that's really the gist of where soul came from. And then once I started growing the mushrooms, I got into the medicinal aspects of it. I noticed a huge demand. I noticed a health crisis in our country. And I started researching and found out that these mushrooms have been used for centuries, thousands of years in some cases. For example, reishi's been used for over 4,000 years in Eastern medicine, known as the mushroom of 100 diseases or the mushroom of immortality, the queen of mushrooms, the spirit mushrooms. It has the longest history, and it's known for its antioxidants, anti-inflammatory, antihistamines, liver health, cardiovascular health, anti-cancer, anti-tumor, regulating blood sugar levels, hormonal levels, adaptogenic, the body adapt to stress, provide an overall calming effect. It also can improve sleep quality, and rich in polysaccharides that regulate the immune system, stimulate T cells, and like I mentioned, inhibit tumors as well. So hence the name of the mushroom of 100 diseases. And then of course, I didn't bring an example today because they're still in their fruiting environment, cordyceps, which if you've heard of The Last of Us, it's a fictional series and a video game about a zombie apocalypse. And that is based off of this mushroom, cordyceps. It looks like Cheetos. There might be some pictures in the slide. And it's known as the athlete's mushroom, provides an acute energizing effect. Just like reishi, it's also parasitic, but to insects, so living organisms that range from spiders, ants, and in this case, silk moth larvae. And they attack the nervous system of that insect, turn it into a zombie. So one example is a species that will attack the nervous system of an ant. Ant will essentially die from that fungus, and that mushroom is taking over its nervous system and controlling it. And it's still walking around and moving. It walks to the top of the tree, to a branch. The mushroom fruits out of the head of the ant, and then spreads its spores and finds another host. Same concept with all the species, but different hosts. And in this case, Cordyceps militaris. Centuries of use for energy, oxygen flow, improving libido, men and women, and adaptogen as well. It finds the host of a silk moth, and it's a larva underground, fruits out of it, only in the Appalachians, so it only can be found in certain regions of the world. And there's only a couple guys in the country that are breeding their spores to create stronger lines of genetics, allowing cultivators like myself to grow the mushroom. Hence why it's super expensive, one of the most expensive mushrooms in the world. Can you go to the next slide? I don't normally use a PowerPoint presentation, but yeah, so general ideas are the mycorrhizal mushrooms creating a symbiotic relationship between the nature. It's a mycelium network underneath us. They are connecting all the plant life, all the trees, and the trees are able to communicate with each other, provide nutrients. They can signal that there's any type of threat in the area, and even mother trees can support their babies with nutrients through this mycelium network. So that's the mycorrhizal mushrooms. They grow on compost or soil, amazing for your gardens as well. And they play one of the largest roles in nature versus the saprophytic mushrooms, which are going to be breaking down trees and other matter. Otherwise, nothing would digest and break down and decompose. And that's the role of these mushrooms here. So the growing environment, which is super crucial, it starts in a lab with a petri dish, like I mentioned, but it ends up in a fruiting chamber with 4 variables that are extremely important to maintain and grow a happy mushroom. And it comes down to temperature, CO2 levels, which get exhausted out to maintain that parts per million below 800. Anything above that, the mushrooms will not grow properly. They'll change their morphology. They'll look different. CO2 oxygen, of course, that goes in harmony with CO2. Light. So people believe that mushrooms need to be grown in the dark. It's not true. The incubation period is done better in darkness, but when they're growing, they need light. They're not photosynthetic, but they're photosensitive. So to develop their pigmentation and to just be a happier growing mushroom, light is super important. So we simulate that process in 12 on, 12 off. And humidity, which is one of the biggest factors, and typically around 85 to 95% humidity. And that humidity is going to signal the mushroom to fruit. So if you've ever seen mushrooms pop up after a heavy rain, it's because that moisture content is initiating that mushroom to start the end of its life cycle, which is reproductive. So the reproductive aspects are their caps here. So this mushroom might be looking to see if there's any spores, billions of spores releasing off of it. And you can't see them in most cases, or sometimes it'll look like a little smoke, but that is the end of the life cycle, and you can grow off of the spores, or most importantly in our environment, we want to harvest them before the spore release because they make a mess. they'll clog our filters. And that's essentially it right there. Those 4 variables, and that fruiting time takes anywhere from 7 to 10 days, or about 2 weeks for lion's mane. And then everything prior to that is done in a laboratory. And then incubation is done in vitro in these bags here. So this is still growing, it's alive. And when we're ready to fruit them, I'll cut the bag off. But that is allowing moisture content to be retained in there. It's about 50% hydration. So we do 5 pounds of water, 5 pounds of oat and soy hull, which comes in a compressed pellet form. And then after the bag is sealed, it gets sterilized for about 14 hours in steam sterilization, which is going to kill off any bacteria, any potential endospores that are harboring in there, creating a perfectly clean surface for the mushroom to thrive on. And then, let's jump to the next one. And of course, it's all organic. Pretty much everything goes back into the soil of the farm. It's a regenerative approach, and I'll share a little bit more about that. But yes, these are nutritional powerhouses, they're medicinal powerhouses. So all these mushrooms, they have essential vitamins, minerals, and proteins. They are a superfood. They contain what the body really needs. I know people that are almost on a strictly mushroom diet, and they're as healthy as can be, but you need to cook your mushrooms to unlock those nutrients. So common misconception, you go to a grocery store and you'll find sliced cremini buttons in a salad bar. You're getting water content out of them, and pretty much nothing more. The cell wall of the mushroom is lined with chitin. It's an abundant polysaccharide found throughout nature. It's what crustaceans actually make their shells out of. But our body cannot break down and digest chitin. So in order to break that down, heat treatment is needed, essentially cooking. And that unlocks the nutrients and will prevent an upset stomach. So an example is if anyone has ever experienced psilocybin psychedelic mushrooms and they can give you an upset stomach, your stomach is not breaking down and digesting it properly. That explains that where you get nausea. So cooking your mushrooms is extremely important, not just for nutrients, but to prevent nausea and an upset stomach. And then there's the medicinal components, which are going to be the mushrooms that we work with. Some of them are culinary as well, but they have a very complex cell wall that cannot be broken down without 2 extraction processes, which is hot water extraction, which is essentially anywhere from 2 to 8 hours of making a tea, steeping it at a specific temperature, and then an ethanol extraction, which is essentially a tincture. And that is going to pull all the non-water soluble compounds out of the mushroom, making them bioavailable to the body. So you could even cook reishi, but it's not going to do anything for you because it needs several hours to break down a really tough mushroom. And they're very woody. You know, nothing's really going to penetrate that without really fine milling it and then starting the extraction process. So the 5 mushrooms that I mentioned, lion's mane, it's one of the most popular mushrooms right now. We do an extract of that, but it's also a gourmet delight. It has texture like crab meat, some would say lobster. It's very meaty. You can manipulate it in many ways, impart flavor onto it, but it's really celebrated for its medicinal benefits, which are 2 compounds called brissinones and erinacines found in the fruiting body and the mycelium. And these compounds are small enough to pass through the blood-brain barrier and promote the synthesis of nerve growth factor. NGF is what our brain produces that supports our cognition, mental clarity, and eliminating brain fog. But as we get older, it produces less and less. And this mushroom is able to create NGF in the brain and support brain-derived neurotrophic factors, which are proteins in the brain and the spine that support the genesis of neurons and the overall health of them. So it's why they call it the brain mushroom or the smart mushroom. And it comes in many different shapes and sizes and forms. And that one on the top left is Hericium americanum, which is found in North America. It has these longer spines. We also grow Hericium erinaceus, which is what extracts are made of, what we most commonly grow because it's a large producer and it has all the medicinal benefits in it. Turkey tail, which that's on the farm right there. We wouldn't use that for an extract because it's growing off of treated wood. But we thought it was worthwhile taking a picture of it because it shows that that mushroom grows everywhere. Known as Tremedi's versicolor because it comes in many different colors, from these bluish, grayish, reddish colors, a lot of different tones. And it's the most researched mushroom in the world. It's been used in cancer treatment for over 3 decades in Japan and China. It is the most rich in beta-glucans, which is the hallmark of all these mushrooms. Beta-glucans are immunomodulators. They regulate your immune system. your immune system can overperform or underwork, and beta-glucans will bring it into that Goldilocks zone. Highest concentrations in turkey tail. And it's also known to stimulate T cells and inhibit tumors in the body with other polysaccharides known as polysaccharide crestin and polysaccharide peptide, which is really why the research started 30 years ago, and now it's being implemented in cancer treatment in conjunction with chemotherapy, radiation, or not. We've had almost a dozen cancer patients come to us looking for that mushroom. I used to have to educate on it. Now it seems like it's made its way into the mainstream where people are looking for it specifically for cancer treatment, and they send us their CAT scans, their T cell counts, and the results are pretty profound. And many of them have gone to complete remission and no more cancer. And it just took time for it to get to the spotlight, and it's probably going to remain there for a while because it seems like cancer's on the rise right now. Cordyceps, which is the mushroom that I mentioned earlier, the zombie fungus. It's one of the most expensive mushrooms in the world. You can eat it, but it's not really worth it because its medicinal benefits outweigh its culinary. It's delicious. It tastes exactly like lobster to me, which is a really interesting experience, but it's going as much as $500 a pound for organic here, and $900 for the albino strain, which we grow as well, which has higher levels of active compounds. But very cool mushroom, amazing history. Studies being done on leukemia and HIV treatment as well. And then reishi, which is one of my favorites to grow. There's many species here, and like I mentioned briefly earlier, we grow in a different environment where all it has is light and a grow tent, and CO2 builds up, humidity builds up to about 100%, and we starve it of oxygen, and that allows us to get different styles, aesthetics out of it. Essentially, the inspiration came from bonsai, where I used to do bonsai and I wanted to express creativity, and I learned that this mushroom is sensitive to light for its pigmentation. It'll grow in that direction of where the light source is, and then I can get different styles out of it, have fun, and then preserve it. So this will get dried probably today, but you wouldn't notice a difference when it's dried or not because it keeps its morphology because of its low water content. But like I mentioned, antioxidants, anti-inflammatory, antihistamines, liver, cardiovascular health, and the list goes on, hence the name the mushroom of 100 diseases. And we grow as much as 7 species, and we narrowed it down to 4 for their higher levels of active compounds. And, chaga, which I have a picture here. It's a non-cultivated mushroom. It's considered a sclerotium. It's a mycelium mass that grows out of birchwood trees. It's really interesting looking. It's like a rock, so processing it is not easy either, but it is the most rich in antioxidants amongst almost any organism on Earth. Great for regulating blood sugar levels, digestion as well, and overall immune system. That's a mushroom we work with foragers up in New York as well, and it's seasonal, so we have to get it when we can. And yeah, it's another immune powerhouse. So the extraction process is extremely important if you want to get the medicinal benefits. I mentioned water-soluble compounds, non-water-soluble compounds. Water-soluble means making a tea. Very simple. We just do it at a very concentrated ratio of mushroom to milliliters of our reverse osmosis water. And then we do sound frequency extraction. Traditionally, the mushrooms are soaked in ethanol for about 2 months, just like most other botanicals, but that can degrade the water-soluble compounds. So we flip the process, and this required a lot of testing and R&D, and we do a hot water extraction first for upwards of 8 hours, and then we do our ethanol extraction with this probe, which is an ultrasonicator. And what it's doing is it's emitting 20 kilohertz of frequency, breaking down the cell wall in under 2 hours rather than 2 months. But then it goes through rotary evaporation. So this is a device commonly used in the cannabis industry to make concentrates, because you can also extract cannabis through ethanol. Same process, and what it has is when we have 250 liters, that's only 5 right there. That was my starting point. And it's distilling all the ethanol out. It concentrates it, reclaims the ethanol, so we end up with 200 proof, 100% alcohol, and then we're left with a residue. And that gets added to the hot water extraction, and then we add 20% ethanol by volume strictly for shelf stability, and that allows us to stand out from other companies that are heavily diluted with a very large ratio of 10 to 1, even 20 to 1. Ours are 5 to 1, so there's a very milky quality to our extract. It's very potent, and then we distill out unnecessary ethanol because people are paying for alcohol when what you want is medicine. here's the line that we have right now. So we do combinations for their synergistic effects, but 5 primary mushrooms, and then we do combos for their synergy. Lion's mane and cordyceps, energy and focus. We have a gut health product, which is ginger, extracted the same way through ethanol. That gets us higher levels of shogal and gingerol, and then we do that with 2 prebiotic mushrooms, turkey tail and lion's mane. We do an anti-inflammatory, so turmeric extracted with black pepper, and we add that with reishi and lion's mane for their anti-inflammatory properties. We have a stress relief that we're releasing soon, adaptogenic mushrooms with ashwagandha, and then just other combinations. We do a 5-in-1 mix just for full body wellness, and people want a little bit of everything, but the idea is to isolate specific mushroom compounds for their specific functions, but also do combinations because everyone has a preference. Some examples. This was somebody with lymphoma. She only took prednisone. She didn't do any type of chemo radiation. And this was about 2 months apart, April to June. And this is just off of turkey tail powder. We do a powder versus, we do a tincture as well for full spectrum benefits, but the powder is going to retain higher levels of specific compounds that are extracted in water. But there's no ethanol extraction done in this, and this is specifically aiming towards polysaccharides and beta-glucans. And this has shown the best results for beta-glucan content, which is 51%. The industry standard is about 30%. So it took a lot of R&D and time to dial that in until we had what we would use is the best product that we could possibly make. And then there's tons of literature out there. The science is coming forward now. You know, it used to be anecdotal, and then Asia was a step ahead of us with research, and now the National Cancer Institute, PubMed, all the big universities have been and still have ongoing studies with these mushrooms for treating the body, the mind, and of course, a good transition into psychedelic mushrooms, which are a massively growing industry. And we are working with FIU. We got the grants for it, and now we're waiting for an EPA license so that we can do research and grow the mushroom legally here, because eventually it's going to make its way into the mainstream in Florida. It's only a matter of time.
I have a question regarding that. I saw the other day that they passed a law that having growing mushrooms is now illegal, like psychedelic mushrooms. I don't know if you're aware of that.
Illegal?
Yeah, they made it like even owning the tincture, the
Extract form?
yeah.
I thought it was always in place. In Florida though?
Yeah, they just recently passed, like, you used to be able to buy it online.
Used to be able to buy spores.
Spores. No, spores, and they made it illegal to buy spores.
Really?
They did?
That's a step back.
When did they do that?
Like a month ago.
Oh wow, because I've always known that it was legal.
No,
Yeah.
now they recently made it.
Because you can get a spore print, you could have even mycelium
Spores
of it,
everywhere.
but once it goes into the fruiting stage is when it's illegal.
Yeah, so the reason they was like, because it has potential to become, or something like that.
Well, that's the intention anyways, of course,
Yeah.
but that's a shame. But, you know, sometimes it's one step back before it's 2 steps
Yeah, yeah.
forward.
There we go.
It's definitely waiting game, that's for sure. But that's Florida, you know. We're behind, but at the same time, there's still a lot of lobbyists pushing for it, trying to make it happen. I think we just have to be patient and bring the research forward, because that will really shed light on what it's doing, which we all know it's extremely medicinal in so many different ways. Don't know if there's another scenario.
I wanted to ask you, do you only make powder of that mushroom variation?
Turkey tail, right now, yes.
Okay.
I have chaga, we just haven't launched the product yet. The concept is that the powders, unless they're dual extracted, they're not going to get everything we want. We do a turkey tail powder because the most prominent compounds in it are going to be the polysaccharides. It has other non-water soluble compounds, but we're targeting specific compounds for the powder. To do a dual extracted powder requires a lot of freeze dryers. Otherwise, it's hard to get it into, because I've done it before, and it ends up more of a jerky style when it's dehydrated, making it hard to process. Freeze dryer will allow it to grind down fine, but it's definitely another step that requires a lot of money for commercial freeze dryers. We have 3, but they're harvest right large, which can fit a decent amount, but to keep up with the demand, to do a dual extract powder is very hard. That's why turkey tail is one mushroom that I would say is good, just powder form, hot water extracted. For lion's mane, if it's not dual extracted, you're not getting what you want out of it. which are going to be those hericinones and erinaceans that support neurogenesis and the nootropics out of it. So if you have a lion's mane powder, you want to check if it's dual extracted. Otherwise, if it's not, you're not getting what you want out of that mushroom specifically. So this is designed, this is freeze-dried as well, so it'll dissolve beautifully in any liquid, has no texture to it, and you can put it in any food as well, and it's most importantly bioavailable. So it's already been extracted, and then it goes through the freeze-drying process. But all the other mushrooms, dual extraction is very important because you're going to get full spectrum benefits out of it. It was done a while ago when we were expanding our product line. Now we're expanding the farm itself. We partnered with Lion Farms. They are the largest worm farm and focus on vermicomposting, so there's zero waste involved. Lion Farms was started by Peter Fideli, who's our partner, and he is extending an invite to the Gardening Club and anybody else that wants to join to come and get a full tour of the farm. It's over 10 acres, vermicomposting, so it's got a specific area focused on worm farm. And it is essentially creating black gold, is what they call it, which is fertilizer. And there are specific species of worms, and they produce amazing poop, essentially. And that ends up becoming a dry worm casting fertilizer, or a tea, which only has about 48 hours. And what they're doing, when you come see the farm, you'll see before and afters. And actually, do you have the other side?
I have the other.
You can pull it up real quick. But it's creating all the nutrients the soil needs, and it's long lasting. So you can treat the farm or any garden one time, and it will remove heavy metals and support nutrients that it needs, nitrogen, phosphorus, potash, for extended periods of time. And the farm essentially has a process where they have these giant vats with about 4 feet high of compost. We're continuously putting mushrooms into it. We're putting the actual substrate blocks into it. The worms are breaking down and continuously eating it, and then the top layer ends up being food. Then right below that is the worms, and below that is all worm shit. And we scrape off the top, we put the worms aside, and then we remove all the castings, and then we run it through a tumbler that removes all the rocks and other debris that we don't want, separating all the poop, and then it gets air dried, and then we sell it as a black gold, essentially fertilizer for your garden, or it's moved into a bubbler where it creates a worm tea. It's not a tea in the sense of drinking it, but it's feeding the entire farm, and the irrigation system on the farm is supplied by this worm tea. And then actually we'll get some before and afters, but
Is there a specific slide you want?
yeah, just roll through. It's a rabbit farm as well. So
Oh, wow.
the rabbit poop goes back into specific vats for breeding. So there's ones that are designated for the worm breeding, and then there's ones designated specifically for making compost. There's zero waste on this farm, so everything goes right back into the soil, into the worm farm, or creating more compost. So there's zero waste, and everything becomes full circle. There's aquaponic program as well. We just put a pause on the cannabis program, which we're doing with FIU. So we have FIU students that are on the farm just about every day, and working on this vermicomposting program and CBD or cannabis in general. You can scroll back one or two. Yeah, so this is the larger facility now, and sterile environment, keeping it clean, and simulating nature with 4 variables that create the perfect environment. The spent mushroom blocks go into composting or back into the worms, and everything just gets fed right back into the farm. It's one of those things where pictures and talking about it doesn't do enough justice, so coming and visiting it will really speak volumes, and yeah, everything goes full circle, hence regenerative or vermicomposting. There's some FIU students right there.
How's up?
So actually that might be so this was before 2021. Let's see if there's an after 2025. Extremely lush. So within 4 years, but it was less than that, the farm has flourished based off of this vermicomposting program.
Oh, that's the cannabis thing you're talking about too? I see up there.
Yeah, that's it. It's Jordan, and that's Daphne, and their students are going to the masters. and we're expanding out on more tissue culture and a couple other botanicals that we're going to work with in the lab with these students here. Cool. I could get onto the science of this, but it's not my bread and butter, so I'll save you guys on that one. But which is why Peter extends the invite to come out, see the farm, come in as a group, and you can reach out to me or Gio, if you want to leave my
Or
information.
John, or I'll.
Cool.
Yeah.
I'm going to leave some flyers here for you guys as well. Check out the Instagram, our website. We cross-mention Lion Farms there. Oh, here we go. I want to go back a couple slides. So that's our, it starts with a culture like I mentioned. This is our cordyceps grows, which is a very cold environment and light. We grow in these bins here. Let's see what else we've got. Lion's mane with different styles of reishi, and then the farm as a whole. And that's Oscar.
Thank you.
You guys have any questions?
So I did about the reishis. So there's different deprivation techniques to get different like phenotypes off of them. Have you noticed any difference in chemical composition between any of the different deprivation techniques, whether it be light, oxygen, carbon,
Yeah,
and
so-
which ones show up more?
Definitely. When it comes to light and CO2, it's going to change its pigmentation, and there's definitely a lot of different phenotypes I work with, specifically of this species. But it came down to just trial and error and noticing that there's changes in their pigmentation depending on how close they are to the light source. And then if there's a phenotype that I like, or phenotypes can change over time, those physical characteristics can be cloned. And then I can essentially grow the exact same mushroom. And over time, you get degradation in the, not necessarily degradation, but changes in the DNA that create mutants. And sometimes a mutant is more favorable. So I would essentially, because like I might clone this one because I love the pigmentation, the morphology on it. So I'll open it in front of my flow hood with isopropyl, a sterile scalpel. I'll take a tissue sample, and then I can grow almost identical mushroom to that. And I'll do that all the time for something I favor. But over time, if I continue to expand out a culture, it could get mutations that I don't like. So it's about keeping a close eye on it and having a library or a master syringe or master slant, which is just a centrifuge, and you can store it for years on end and always go back to that exact, those exact genetics, because it's a favorable in its morphology and its pigmentation or whatever the case is. So storing it for long periods of time will allow you to always go back to that original genetic that you're using. Did that answer your question?
Yes.
And there's some I wish I did clone in the past
Can you go to
because I
slide
can't
6
replicate
in
them,
the
but that's the name of the game
presentation?
is
Which one?
finding something you
Slide
like and
6.
then playing around with it. A lot of, I'm self-taught, so it all came from trial and error and failing. That one was really cool as well. Is that the one you wanted to see? Oh, I thought you were
No, I
asking.
wanted that one.
This one.
Yeah, thank you.
I also took pictures of everything too. If you need any pictures or something.
I could also send out the presentation I have both.
Hmm.
In summary, and then we share a lot on our Instagram. Our website has some information about the extraction process. Transparency is really important. So if you scan the QR code, it'll take you to our COA, our certificate of analysis. It'll show what our active compounds are. There's a lot of snake oils out there, and they're getting exposed. And we were just dead set on making sure that we had a tried and true product, that it contained mushroom in it, and we do ongoing testing to show forth that we're honest, but also you're getting actual mushroom here, versus a lot of companies have little to nothing in their extracts.
I have a question. So what kind of fume hood do you use and what's your contamination rate?
So we use a standard flow hood, so it's a HEPA filter. We're about to expand into our new lab. It's going to take some time. We're going to have 4 2x4 flow hoods. We don't have a fume hood except in our extraction lab. We're building out another one to exhaust any fumes out, but right now we're just working with ethanol. And what we're moving into is going to be in the cellar of the grow room, and it's a lab that's going to have positive pressure. Air that comes in is passing through a UV light sterilizer, then through a HEPA filter into the lab. And then that lab is going to be, production is going to be done in front of those 4 flow pits. And then, yeah, just balancing the pressure because there's going to be clean air coming in, and then everything will go into an incubation room. Contamination rate is about 2%, but our current flow hood is starting to die out, and before we just buy another one, we're just getting ready to buy 4 new ones to put into the new lab. And then sterile procedure is super important, so I'm training people in, and I notice contamination increase when I'm not in the lab. And that is just because people don't have the full scope of how sterile you need to be. Isopropyl, every surface, your arms, your gloves, your lab coat, everything needs to be as sterile as possible. Otherwise, I assumed a spore was on me or in my hair, and I grew another edible species, and in harmony, surprisingly, reishi grew out of it as well, out of the substrate block. So you can't see the contaminants. So even wearing a hairnet, a mask, a lab coat, and then gloves and isopropyl on pretty much every surface. On top of that, it's just about working in front of a HEPA filter so that you have clean air coming through. And of course, that has to be monitored and changed every so often, but you don't really know unless you put a lighter in front of it. You can see the airflow. Otherwise, there's contamination you just can't see. But it was about 2% for about a year and a half, and as other people come into the lab, that's when you see the increase. So, no one's going to ever treat it the same way as you do when it comes to your business or just your livelihood. So, you can only do so much when you're not there, but you can teach people proper sterile procedure, and eventually they'll catch on. But it takes some failing to really see where all the faults come into place. Any other questions?
Just curiosity, on one of the last slides for the Lions Farm presentation, there was an orchid growing with the golden oyster mushroom. The orchid being a saprophytic plant and the oyster mushroom being saprotrophic, are they able to grow
Yes.
in that sphagnum moss
They could.
solution?
I didn't see that
Or was
slide.
that just like a...
I don't know. I
Is
don't
that
think
just aesthetic?
Yeah.
Yeah.
I was
Just
gonna
a
ask the same
stump.
thing.
so. Yeah, I can imagine. I mean, a mushroom would grow in harmony out of a tree, especially a saprophytic one with an orchid. I don't know what you're talking about. To be honest.
Yeah, it was like
I think it was the last one. Yeah, there we go.
Oh, yeah, that was aesthetics.
That was,
That wasn't
yeah,
that.
sure,
Yeah.
yeah.
Yeah, it's a cool.
Well,
ice cream.
I was assuming, but I was like, that's super cool if that's something that you can
It's worth
You
trying.
should just
and
grow
experimenting.
orchids on anything. You just
I've seen people
Orchids
grow it
are
on.
super finicky.
Yeah,
Yeah,
so
but depends
sphagnum
on what kind
moss
of
or
orchid you're
pine
talking
bark
about.
you can grow them on, and I don't know if that pine bark would be too large to inoculate.
Pine is not the most favorable with mushrooms. I've seen some guys have success with it. I think in New Zealand they don't have access to oak, so they end up making it work for pine. But it'd be a fun experiment to play around with. But when it comes down to competing with another organism, most likely the other organism is going to outcompete the mushroom, just because mushrooms are even more finicky than pretty much any other botanical that I've ever experienced. So yeah, that's just aesthetics. It looks cool, but that's not.
Yeah.
And breeding mushrooms too. You're saying you have a lot of people that are going out to. How would you enact that? And a lot of times these mushrooms are like asexual and kind of just have their fruiting body and produce themselves.
So there's 2 guys with cordyceps. So cordyceps, which is the orange Cheeto-looking mushroom, it is prone to what's called senescence. So the genetics will degrade over time in a short period of time. So there's only a couple guys that I know of, one of them being one of the leading mycologists in the country, Ryan Paul Gates of Terrestrial Fungi, in Mississippi, I forgot where he is, close to the Appalachians. And they go out and forage for them. They find new specimens, and then they'll, I don't know their exact process. A lot of it's proprietary, but you could leave the mushroom on like a piece of aluminum and it'll spread spore there. How they specifically do it, I don't know, but they'll isolate the spores and they get them to breed over time, and it creates another line of genetics that ends up being stronger and less prone to senescence, but it will still degrade. And that's why these guys have proprietary recipes for storing them longer. I understand why they wouldn't share it, because it keeps them in business. And it's their mind that created this. Without them, I wouldn't be able to grow cordyceps. The exact process for breeding their spores, I don't know, but some of the reishi species that I grow are made off of breeding different spores, trial and error, until they found a perfect specimen. So the Ganodermas, one of them that I grow, I should have brought it, Lincoln number 7. Number 7 meaning it was the seventh out of 10 different breeding pairs that he created that had the longest antler style. So he stuck with that one, and he probably has the original culture from who knows how many years ago. I haven't gotten deep into breeding. Hopefully I get the time, but the downside is that we don't have the luxury here in South Florida to find really nice specimens because most of them are going to grow north of Florida. You can grow some Ganoderma species here. I found them, not the most medicinal. And then flamingo chanterelle, during the right time of year at the right place. If you're familiar with chanterelle, that's a very desirable culinary mushroom. I found them by Matheson Hammock when it was slightly cool out just after rain. Aside from that, I've never found an oyster here in Florida. I know that there is a Florida oyster, but more north of us. So we don't get the luxury of going out foraging or hunting for mushrooms and then playing around in the lab, except for maybe some Ganodermas. Turkey tail, it's so thin that it's really hard to clone that mushroom. Its cap is like almost paper thin, so the best way to do it is to get a really large harvest, and you can get into deeper tissue to clone it, or some guys will fresh mushroom that hasn't sporulated or just started to, you put it in a sterile, you could use like in a flow hood or a sterile box, aluminum. You could just cut the cap off the mushroom, let it sit there, and after 24 to 48 hours, it'll create a spore print. And then you can use that for breeding, or you can get a sterile swab, swipe that spore print off, put it onto sterile agar, and throw it out from there. But I find it easier to just clone a tissue sample as deep as possible and get it onto many plates, because the contamination rate is always going to be higher when you're working with a clone or tissue sample, because wild forage mushroom, or not wild forage, is going to have all kinds of contaminants on it that you can't see. So I'll spray it down with isopropyl completely, then split it open or cut it open with scissors, get the sterile scalpel, take as deep of a tissue sample as possible, get it onto agar, and pray that there's no contamination. But if there is, in some cases it'll slightly outcompete, or when you see the first sign of mycelial growth, and sometimes you'll get cobweb or slime mold on there, you can isolate that mycelium that's slightly away from the other contaminants or the molds, put it onto a new agar plate, and 9 times out of 10, I'll get a clean tissue sample or a clean mycelium sample and continue to expand it out. Any other questions, guys?
You had mentioned that, I mean, I might as well. You had mentioned that, like, in nature, a lot of times they have their own, like, immune system with, like, beneficial symbiotic bacteria and everything. Is there any way to isolate those to have, like, in a controlled setting to go along
With that
with the
immune
fungi?
system?
So,
No.
no.
Yeah, otherwise someone would have probably figured it out. Think of War of the Worlds, where the extraterrestrials came down here, they invaded, and their demise was the bacteria that we live with here. And we live in harmony with it, but because they're not of this earth, they could not outcompete it. It killed them all. When you take a mushroom out of its natural environment, where they've survived for eons with other bacteria, it no longer can withstand. Why specifically? I don't know. It's just their immune system changes. Even if you wild clone one, it's just everything changes once you bring it into a controlled environment, a laboratory. And that's why the laboratory is so important, is to eliminate contamination, which will happen. Everyone gets contamination at some point. You can only do your best to mitigate it.
For the reishi that you play around with, you said like you used to do bonsais. Do you play with the color of the light or just the position of the
Position. So I use full spectrum. It seems to just keep them happiest. I know guys that would use like full spectrum red and blue and purple, and notice no difference. In fact, I've seen grow operations entirely of that, more of like the purple or red and blue, and their mushrooms come out identical to mine. So not necessarily the type of light, but the position when it comes to reishi. So this one was right next to the light like that, which allowed me to grow it outwards. If you check on the Instagram, I have ones that have, total like almost 90-degree angles. This one was below the light but off-centered, which is why I kind of grew that waveform pattern. And then this one, same thing. You could tell where the light was positioned based on where it blew, versus this Ganoderma was just directly under the light. So it's just playing around with it and having fun and figuring out what style I like and how far I can go with the manipulation. And then the light can change the pigmentation. So I've noticed some of them directly next to the light have brighter colors than ones that are at the bottom of the road. And I kind of figured out there's no like book on it, anything like that. So, but that
book.
Good. Found that idea.
So just based on observation, you start to see patterns of like which ones get darker, I guess, from what you just explained.
So, I mean, if you look at my Instagram, there's one I just posted, or my social media posted, manager, and it's the same species, same exact genetics, and it has a much darker pigmentation. And that's because I started it lower and then put it far away from the light. But yeah, and I bet the lux on the light could also affect it too. So your environment might not grow in the same as mine, the mushroom itself in that environment. So you could learn from somebody, start your own process, and then you'll get totally different outcomes. But that, I think, makes it more exciting. Then you get to figure it out on your own rather than have someone just give you the cheat sheet. Anybody else?
Are you selling the tinctures?
Yeah. Always selling them. We have our website as well. We're at right now 4 farmers markets: Pinecrest on Sundays, Palmetto Bay on Sundays, Coconut Grove Saturdays. We're at the zoo. We're not going to really do it anymore. We do one-off ones, and then Coral Gables when it's in season. The website has all the information. I have products on hand, and then if not the majority, but a good amount of orders we get online are just natives that just don't want to drive. So it's always a way to get it. We do shipping every other day or so.
I bought one of them at the
Society
Charity run,
yeah. I
We're
bought
actually
one.
going to do another one
It works
November
good.
2nd. So
November
in
2nd?
Hollover Park, I don't know if you guys know that. We just got the reservations, and we're going to start selling
I'll note
tickets.
that down.
Yeah, let us know.
Okay.
And then I highly recommend coming out to the farm and seeing everything firsthand. Pictures just never do enough justice. I'm not as versed in the vermicomposting because my area of expertise is in the fungi area, but it all goes full circle. So everything I grow goes back into the soil, into the worms, the cordyceps, the parasitic fungus. We grow it on brown rice. We're running trials right now with chickens to see, after we dry out the substrate, feed the rice to the chickens and see, I don't even know if it's anecdotal or not, but the cordycepin, which is one of the active compounds in cordyceps, makes its way into the yolk of the egg. So it's going to help the chicken itself with its immune system, maybe oxygen flow, respiratory, stamina, but create a medicinal egg out of it. And we're going to run some trials with controls and variables and see what the outcome is, send it to the lab, where they'll test the egg yolk and the whites and see if there's any medicinal benefits in there. So no waste.